Okay, let's start the class. Today will be a review section. So first of all, the same thing, we want to go over it. All these material underneath the eCampus. So basically we have, I put slides first. Then will be linked in Zoom. It's a Zoom link slides video. And then it's a classroom teaching video. And everything is the order like that. So these are the materials you can go back to review if you missed something. Okay? If you missed something, then you can go back to review. This one I pretty much need to be moved up to here. But I can do it real quick. Okay? And that's good. Okay. Okay, now, then we have all these questions for you. It's a practice exam questions. And the real exam questions will be pretty much the format and the content will be very similar to this. And the level, the difficulty level is also will be similar to this. And I hope you're going to do it already, or you haven't done it, please do it. It's like a practice by yourself, and the key will post it on eCampus tomorrow. Then over the weekend you can take a look, and next Tuesday right here will be the lecture exam 1. So let's talk about these questions. I would love to do the review is combine with some of the knowledge together, not only a single piece of the sporadic knowledge. I want them combined. So very important here we talk about is the STEM technology. And among the STEM technology, we practice in the lab. We have a small unknown number 1 for you is the gram stem. And we also going to have some questions in the lecture for you. So gram stem, we talk about the procedure is crystal violet. Okay, the function of the crystal violet, you should know it's a basic dye. And then you have a second step is using gram iodine. And the gram iodine will make the crystal violet stay strong. And then the third step is 95% alcohol, is decoloration. And basically at this moment, the slides ideally should be blank or nothing, or it's crystal violet still there. The last step is saffronin. And the function we call it a counter step. And this is a very simple 4 procedures, which is certainly answers the question in this short answer. If you look at the short answer, is question number 21, describe the gram stem procedure and briefly explain the function of each step. That is, you answered this question already. Now, we have some multiple choice questions, so you can see what it is. Let's look at question 70. If the decolorizing is left on too long in the gram staining procedure, gram-positive bacteria, gram-positive organisms will be stained black and the gram-negative organisms will be stained. So that's a question for you. So right here, if you see, is this guy stains too long? I say this is only 10 seconds. If this guy is way too long, let's say it has been decoloration. Decoloration. An hour, let's say, just make it a little bit, stimulate it a little bit large, okay? 1 hour, what's going to happen? So what is the answer? Then gram-positive organism will be stained what? I want to have you guys have some reaction. What is the answer? It will stay the pink, is that right? Because it's all gone. So crystallization is gone. Then you counter still with saffronin. It shows the saffronin color. Is that right? So the first one is pink. How about the gram-negative bacteria? It's not going to affect it because anyway, it's going to not hold on. Is that right? You still need another hour, it's never going to hold on. So, it will be the pink. And what's the pink of the color? Is saffron. So it's pink, or we say it's red. Okay, so that's a question, we solved the problem. Let's look at other questions, whether we have related to the gram stain. Let's look at these. If there is some other question related to the gram stain, we still can figure it out. If we don't have it, okay, if we don't have it, let's jump to this question. So we talk about the gram stain. Okay, gram stain has two theory, which is cell wall theory. And the lipid theory. And we already mentioned, so I'm not going to repeat it all the time. So the question is, if I give you a diagram, looks like here, can you do a label? Can you do a label? So here is the question. Clearly label the terms may be possibly applied, but not all the terms will be applied. The terms is, first of all, we're going to ask you gram positive or gram negative. Outer membrane, inner membrane, peptidoglycan, lipopolysaccharides, periplasma, transmembrane protein, teichoic acids, cytoplasma, and the lipopolysaccharides. You label hydrophilic and hydrophobic. So first thing first, is this gram positive bacteria or gram negative bacteria?
Great.
Positive or negative?
Negative.
Yeah, it is gram-negative bacteria. Then why? There is one obvious reason. The peptidoglycan is very thin. Is that right? So, you can record gram-negative bacteria, peptidoglycan is very thin. Now, obviously, as a gram-negative bacteria, the existing LPS lipopolysaccharides is there. So, how are you going to do the label? When you do this, on the top, write gram-negative bacteria. Is that right? So, gram-negative bacteria we will be choosing. So, you can mark it anyway like a red or something. Now, of course, gram-positive bacteria you're not going to pick, so you move them away, cross it. Is there outer membrane? Yeah, this is outer membrane, is that right? Is there inner membrane? Yeah, that's inner membrane. So these are you're going to have to label. When you label these, be careful. If you label, okay, let's say this is a bacterial cell wall, okay, you label. If we have this question in the exam, please label it very clearly. This is outer membrane. Very clearly, okay? Then you know phospholipids. Very clear. And this guy right here, very clear label, this is hydrophilic. Then go down right here, hydrophobic. Everything is very clear. People can recognize it. Okay, and then write your handwriting slow and clear. You don't say, oh, somewhere outside here, outer membrane. There's somebody do like this. Who knows which one you are doing? Or so somebody like this, phospholipids. If I see these two answers, which means I don't think you understand where it is, okay? So these two avoid it. very clearly to label where it is. Okay, that's a key. So I'm showing you already. Now let's find these. Okay, outer membrane. Do they have peptidoglycan? Of course, is that right? Because it's very thin. That's a very thin one right here. Lipopolysaccharides on the top, right here. Okay. Periplasma, do they have periplasma? Yeah, where it is. This problem is you're gonna have to draw the whole thing. Probably. There probably you have to write a big blanket to draw. That's a periplasma. Because there is a two there. Transmembrane protein. What is transmembrane protein right there? Is that right? How about on the top? On the bottom? Yeah. Teichoic acids. Do they have teichoic acids? No, because teichoic acid is in gram-positive bacteria. Do they have cytoplasma? Yeah. The cytoplasma, you have to draw it again. Okay. Lipopolysaccharides, we already tell you about it. If you don't know about this, let's say you're still confusing, what you should do, then go back to look at your slides. The slides will be pretty much is right here. This is what's the reason for these practice questions for you. And you can have a study group. Just in Evansdale Library, some room, just find some boys and some girls, just study together. Spending 2 hours, you solve all the problems. I'm telling you, go here if you don't know. Is this one over there? Plasma membrane, gram-positive bacteria, if you don't know. Is that over there? So these two slides, which tells you what's the structure of gram-negative and the gram-positive bacteria. Okay, so that's something we will talk about, is labeling. Okay, so we finished talking about this question. Then we go back to see the multiple choice. They may have some question related to this. We mentioned about the lipopolysaccharides right on the top, LPS. Then we have a multiple choice for this one. So you go here to see. There is one question related to lipopolysaccharides. The lipopolysaccharides that is found in the outer membrane of gram-negative bacteria is also known as what? Do you remember right here, I wrote the notes, I said LPS is a major issue why Salmonella, E. coli, and the gram-negative bacteria will be a problem because they will generate endotoxin. Is that right? So the answer here is pretty obvious, is D. Okay, is dog, is D. Question 29. Okay. So once we did this, we talk about the cytoplasmic membrane. There's 28, there's a question for that. The cytoplasmic membrane of bacteria, what is belonging these answer will be belong to that. Retain the cytoplasm and its contents. Acting as a selective permeable barrier, allowing some molecules to pass while prevent the movement of the others, is a major site of ATP synthesis in errors. What are the choice? Is that ABC is all correct? Yes, that's all correct. And if you don't know, then you should go back. We have slides, the cell membrane slides, which is talk about all these answers. So these practice questions is give you review material to go back to read the slides. The slides here, which is tells you what the answer right here. Then you can read through these real quick. Okay? So that's a question we talk about. Okay. Then, when we talk about this guy, we have some questions for the other methods to do the stain. We talk about the stain technology, for example. Let's do this question. question 15 and 16. So we all know that we have to do air dry and heat fixing. So prior to staining, smears of microorganisms are heat fixed in order to. Why we have to do? Allow eventual visualization of internal structures. That obviously is not good because light field microscope, we won't see the internal structure. Basically is a laser confocal or sometimes is a phase contrast microscope, we can do that. Or like not a scanning transmission electron microscope. So that's not A. Ensure removal of dust particles from the slide surface. You just simply clean it. You don't have to do it like that. Attach it firmly to the slides. Is that correct? Yes, that's correct. So the answer is C. Then create small pores in cells that facilitate the binding of stain to cell structures. That's not correct. Okay, the next question, 16. Acid-fast organisms, when we give the example, microbacterial tuberculosis contain constructed from mycolic gases in their cell wall. Where is mycolic gases will be? So first of all, acid-fast stain. We have, the reason is we have a very waxy cell wall. The major components is mycolic acids. So where did this come from? The question is. Come from protein, carbohydrates, lipids, peptide, or glycan. Where they come from? And I wrote that. I said it's a long, unsaturated fatty acids. Okay? So the answer here is lipids, is C. Let's look at the question number 18. Basic dyes such as methylene blue binding to the cellular molecules that are. This is go back to the question, very simple. The bacteria, when they grow, how they charge them? Is that negative charge? Now all those dye, how they charge them? It's a positive charge. Okay, so they can combine together. That's what we did in the lab. So these are the dye, positive charge. Now, of course, it's a basic dye. If it's acid dye, you're going to charge the background. That's dark field microscope. Okay. So, basic dyes such as methylene blue binds to cellular molecules that are, that's English, that are. Cellular molecules should be negatively charged. Is that right? So the answer is B. Let's look at this question. 19. Because transmission electron microscope use electrons rather than light, it is not necessary to stain biological specimens before observation there. True or false? Yeah, it's false. Why? We still have to do the stain, but we call it a coating. Is that right? Silver metal to do the coating. So the answer is false right here. Okay. Small internal cell structures are best visualized by, that's question 20. If you have a light microscope, dark field microscope, transmission electron microscope, flagellar microscope, we don't have flagellar microscope, it just made up something to confuse you. What are the one to see the internal structure? That's TEM, is that right? Transmission electron microscope. The answer is C. So these are the questions which is gave you a bunch of that to talk about the basic of the microscope. Okay, now let's move on to the question. One of the questions I really want to talk is question 37. This is the question I want to talk. If you can try to do it by yourself, you can do it. We will do this in the lab real quick after next week. So question 37 is mannitol salt agar only allows the growth of halophiles. So what's the definition of the halophiles? Is salt-loving microbes. Non-halophiles will not grow. Among the halophiles, mannitol fermenters will produce acid that turns the pH indicator yellow. Mannitol non-fermenters leave the medium red. Onto MSA agar, you inoculate the halophilic mannitol fermenter and the halophilic mannitol non-fermenter. In this case, the medium is acting as what? So let's talk about this. You can think about this. This is mannitol salt agar. Mannitol salt agar has two ingredients. Number one is salt. They are containing 7.5% salt. And we talk about that. Most of the bacteria will not be survive in the high concentration of the salt. So we call it a non-halophile. And the only one which can survive, which is a halophile, and I said majority of the bacteria, which is kind of like Salt Lake City, deep Salt Lake City, in Salt Lake, Great Salt Lake. Some of the bacteria really can survive is Staphylococcus aureus. And also we say Staphylococcus. That's very easy to understand it because Staphylococcus could be grow on the skin surface. And your skin has some salt. It's the first defense system of your body. So salt is a selective ingredient. How about mannitol? Mannitol is a sugar. So bacteria could be fermented or non-fermented. If mannitol fermented, it will turn yellow color. Because we have a pH indicator. The pH indicator there is phenol red. The phenol red will turn yellow. And if it's mannitol fermenter negative, it will be colorless or original color. So what's the function of mannitol? It's about chemistry reaction, is that right? So mannitol here is like a differentiating ingredient. And here is the selective ingredient. Now you have to read the question. If I'm growing a halophilic fermenter, mannitol fermenter, and a halophilic non-fermenter, then at this moment in this media, what's the function of this media? What is your answer? If the answer is C, why we have to tell you the whole story then? I'm growing two bacteria. One is a halophilic mannitol fermenter. Another one is also a halophilic mannitol non-fermenter. If you pick a C, is that right? You think it's a selective media and also a differentiated media. What do you mean selective? Which means one of them won't grow. Is that right? If you have a halophilic and non-fermenter, mannitol fermenter and a halophilic mannitol non-fermenter, both of them will grow. So the media at this moment is not going to act like a selective. The media at this moment, you will see the color difference of the colony. One is yellow, another one is colorless. So what's the function of this media? It's a differentiated media. Is that right? Yeah, that's correct. How about if I change the question? If I change a halophilic mannitol fermenter and a non-halophilic mannitol fermenter, then what's going to happen? The answer will be selective. Because at this moment, this salt is a selective ingredient. The only salt-resistant bacteria will grow, only halophilic will grow. Is that clear? So you have to read the story that tells you why It's not just naturally think about its C, because mannitol salt agar for itself is a selective and fermented it is a selective and differentiated media, but when you're facing different bacteria, it will be different. So I give you a very easy example, you can understand it. Anyway, we will talk in the examinations too. If I have Staphylococcus aureus, and I have Staphylococcus epidermis, I have a mannitol salt agar. What's going to be the situation? Looks like Staphylococcus aureus will grow yellowish color. We'll do this in the lab. You can see it. Will grow yellow. Yellow colony. And the Staphylococcus epidermis will be kind of like a original color. So we say it's a colorless. So at this moment, this mannitol salt agar is a differentiated media. It's very easy to differentiate clinically Staphylococcus aureus and Staphylococcus epidermis because this is a pathogen, this is a non-pathogen. That's why it is important. Okay, if I have a Staphylococcus aureus, then I have an E. coli, what's going to happen? Then the Staphylococcus aureus will grow, it's yellow color, and it will grow. And you will not even see E. coli there. And it's a selective media, because E. coli cannot survive in that high concentration of salt. That's 7.5% salt. Okay, so that's an example. We'll do this in the lab, so you can see it. We will split mannitol salt agar into 3 different cultures, and you can see the difference between that. Okay, very good. Then regarding the agar media, we want to have several questions, and you can go ahead to practice. Let's look at 36. Medium containing some ingredients of unknown chemical composition is called. Remember, we talked about that. If the media containing chemical ingredients is clearly defined, which means every chemical is the formula has been spelled, what is that called? It's called defined media in the synthetic media, is that right? Other than that, it's called complex media. So this is the answer is B, it's very obvious. There is no definition said undefined media. This is just confusing, okay? And this question is also easy. Defined and synthetic are the same thing. So if you choose D, you will pick C also. But there are only one answer, so it will be B. Okay, so that's an easy one. 38. All fastidious microorganisms require which of the following for growth? So which agar using for grow fastidious microorganism is chocolate agar? So this is easy, extra nutrients such as chocolate agar. That's what I added. I think it's too difficult, so that's why I've added blanket, such as chocolate agar. Okay, so the answer is C, very, very obvious. Now here is a question. 39. You would like to culture a bacteria that is growing in the lung of a human patient with ammonia. Which media would be most used in culturing this organism? Transport media, mannitol salt agar, McConkie agar, complex media supplemented with whole blood, none of the choices. Which one you will be picking? Extra 1 point. Of obvious, a little bit difficult. How to do this question? This you need to go back to the slides. What we talk about is right here. When we talk about the bacterial media, I said that there is one slide is very important. You should remember, we will mention it again, is this guy. Enriched and differentiated media, blood agar differentiate for hemolysis. We have Streptococcus ammonia and Streptococcus pyogenes. If you have ammonia, which agar you should be using? It's a blood agar. Because on the blood agar, you can differentiate alpha and beta hemolytic. Based on the ammonia, we have some of them alpha hemolytic and some of them are beta hemolytic, and some of them even non-hemolytic. So blood agar is a good choice. And the blood agar, of course, can make with different resource. So the best answer is complex medium supplemented with whole blood. But I want to tell you one thing. This question is not very good. The reason is that people who make up these questions doesn't have a basic knowledge in the clinical experiences. And if I do, I will choose A and D. Because if ammonia, if it's microbacterial tuberculosis, you also have some ammonia symptoms. What are you going to do? You may going to have to use transport media. You have to send to the reference lab, so transport media might be a choice. So the year when I graded, if you write A or D, or A and D, as long as you touch A and D, I both give you an extra 1 point. This question, when they made it, they didn't understand it. They shouldn't put A there. They try to confusing you, but when you're confusing, I don't think they understand the clinical stuff. The transport media is needed. That is you usually transferring. Because the bacteria is difficult to grow, takes longer time, or we say the generation time is very long, so we need to transport to the reference lab. The reference lab down in Kentucky is in Lexington. I don't know here, it might be in Lexington too. There's only one reference lab. Another one in the south is in Atlanta, Georgia. So it's a couple of reference labs. They handle the final identification. Okay? So this is easy. Blood agar is both a differentiated and enriched media. Yes, of course, it's true. Okay. Okay. We have these questions. I'm going to give you this puzzle to do it. 42. Give a log phase bacteria culture is 1 multiplied by 10 to the 6 cells per ml. And the generation time of 30 seconds, how long does it take the culture to reach a density of 6.4 10 to the cell per ml? This will be a math question. Even you don't understand microorganism, that's fine. That could be an elementary school kangaroo math competition question. It's completely math, okay? First of all, you need to know this. Because when you talk about the generation, it's doubling time. 1 becomes 2, 2 becomes 4, 4 becomes 16, 16 becomes 32, becomes 64, go all the way up 128. Is that correct? Yeah. Then 9, 512. Oh, sorry. This is the easiest way to do it. It's not really smart way to do, but it's okay. 6, 512, 10, 124. This is the first thing you're going to write on your sheet. Okay, how many bacteria are there at the original? 10 to the 6. How many bacteria at the end? 6.4 multiplied by 10 to the 7. Is that right? Okay, you need to know this guy is 664 multiplied by 10 to the 6. 1 digit to move. Okay, so this is the same. So that means 64, is that right? Go ahead to find it. How many generation time? Is that 6? Okay, so 6 generations. What is 1 generation time? It's 30 minutes. And you need to know 30 minutes equals half an hour, okay? So how you do it? Multiply that, right? Equal what? 3 hours. Is that a math question? I think you're not very good at math, that's why I get an extra point of 1 point. So the answer is C. So you have to know this first. Okay, write on the sheet like this. Okay, that's a good one for practice and thinking about. It's testing you about the generation time of 30 seconds. So generation time is equals doubling time. This is important. Equals doubling time. And this means right here. Okay, that's doubling time. Then the question 43 is simple. Fill in the blank. What is the length of time it takes for a population of microorganisms to double in number? It's generation time, is that right? If you don't know about it, you want to go back to review, then you should review right here. That is what we talk about the generation time. And that's a formula is complicated. That's obvious. You have to review the phase of microbial cell growth. Lag, log, stationary, and death phase. When you label, don't forget about there is x-axis. The time and the y-axis is a population of the bacteria. Lots of you, when you label, you don't even label what is x-axis, what is y-axis. That has to be clearly labeled. Okay? So that's a question. We can do the review real quick. Okay. There is a bunch of questions right here. Is we are talking about is external factors to impact a bacteria growth. Organisms that grow well at 0 degrees Celsius and have an optimal growth temperature at 15 degrees Celsius or lower, what is this called? Then this is the question. You have to go back and remember these slides when we talk about the temperature. What's the range? Cycrophiles. That is a bacteria usually is an Antarctic area of the deep ocean. And the cyclotrophs is usually the bacteria can grow in the refrigerator or cooler. Let's say Listeria monocytogenes. Okay, so the answer is cycrophiles. This you have to know the range of these temperature. Organisms that does not require oxygen for growth, but grow better in its presence is called what? Is facultative. Is that right? We talk about aerobic, anaerobic, facultative. Then I said you can learn allotolerant and microallophilic, but we don't want to confuse you. So that's the answer is facultative anaerobes is A. 47 is a little bit easy. Most microorganisms maintain their internal structure, internal pH. How they do it? Of course, it's near neutral. Is that right? Near the pH 7. That's the best way they can grow. 48 is easy. Complex microbial communities that grow tighter and adhere to the surface are called what? What is the bacteria is on the surface? It's in opposite to the bacteria in a broth medium. In the broth medium, we call it a vegetative cells. On the surface, it's biofilm. And where it corresponding to the slides corresponding to is right here, the biofilm formation. That's reversely attached or conditioned surface. And this is a condition requiring for the oxygen. Okay, so you can look at here. So then another question comes out. Organisms that ignore oxygen grow equally well in its presence or absence are called. That's the answer is easy. Allotolerant, because we have a slides here talk about that. Allotolerant anaerobes grow equally well with or without oxygen, which means it's ignore oxygen. Okay. Now, why the reason? Because there is have SOD, but don't have catalyst. Okay. If I have a question I want to ask you, I say, why a Clostridium? cannot grow in the presence of oxygen. And what you should tell me? Lots of you tell me it's anaerobic bacteria. Yes, it's obviously, but you should tell me the story because there is no SOD and a catalyst, and they cannot survive in the presence of oxygen because they cannot neutralize those toxication material. Is that right? So that's a whole story. You should know why it cannot survive. Not only the simple answer said it's an anaerobic bacteria. Okay, let's go to see all these. Okay, next there's a bunch of questions related to the bacteria, cultivation of the bacteria. So let's look at these. Question 52. Samples from a barren river lake were enumerated by both the pore plating and the spread plating procedure. If fewer colonies appear on the pore plating than the spread plating, it is likely that A, B, C, D, E. What is the likely reason? So what's the difference between pore plating and spread plating? And we mentioned about the spread plating is you put the bacteria on the surface and you spread. What is the pore plating? Is your pore melted agar? What's the temperature of melted agar? 55 degrees Celsius, is that right? So this is what you should know. Melted agar. The agar melted, the temperature has to be around 50 to 55 degrees Celsius. This is what we call as a mild heat. And we will do this in the lab following next week. You will feeling that sometimes the bottles is even very hot. You need to wear gloves to hold them. Bare hands you are feeling hot. This mild heat will killing a little bit of bacteria. But spread plating, the agar is already dry, is already solidified, so the agar is at room temperature. So therefore, I said, always pore plating, you get a less number because mild heat will killing the bacteria. So the answer here is obviously heat-sensitive microbes will kill. So the answer here is B. Okay? This is the one we tell you a story. But the story is really we want to know the difference between pore plating and the spread plating. What's the procedure of the difference? That's a key point to test you. Okay, those are the barren river lake is just one too confusing. The idea is the difference between the pore plating and the spread plating. Okay, regarding this, let's look at the question. Look at this question. This is the one we're going to review, simply review the growth curve. The growth curve, we have a lag, log, stationary, and dead space. So, which culture has the longest lag time? That's easy. Culture number 1, is that right? The lag time is longer. Because culture 2 start to grow even. And culture 3 is pretty much steep. Which culture has the fastest growth rate? Yeah, culture number 2, because they are doubling time. Or we say it's a log phase is very short. Which culture has the shortest doubling time? It's still culture 2 because it's very steep. Which culture has the highest cell density? That would be culture 3, is that right? Because absorbance is higher compared to the others. So it's a very easy figure results to tell you the idea of the growth curve. Okay, let's go over the other questions. Let's just go over these real quick. Give the function of endospores and name the two general name in which endospores are generally found. We know that already. There are bacillus and clostridium. So what's the function of the bacterial spores? It's a protection function. Is that right? So why the protection function is there? Because calcium diproconic acids. Calcium DPA. So if you forgot about this, you want to do a review, we could also go back to see your slides. We talk about his last slides right here, which is clearly tells you Bacillus clostridium is 2 genome, has endospore, and the endospore is resistant to the environmental stress. So it's for protection. And why? Because right in the middle is calcium diproconic acids. So this slide is very concise to solve the problem of this answer, of this question. Okay? It's good. Then we look at that. What's the difference between magnification and the resolution? What is magnification? Yeah. How you enlarge the size, is that right? So this is the one we want to emphasize one more time. Sometimes you, magnification. You can easily say this is enlarge size. So that gives you a very simple, is 10x multiplied by 10x equals 100x. Ocular length multiplied by objective length. So what is resolution then? Resolution is a capability to differentiate two closely attached cells, you can say. Therefore, we had a resolution power calculation is wavelength lambda divided by 2, and Na is the numerical aperture, and this is a wavelength. Is that right? And we mentioned if this is low, is better. The resolution power value is lower, but the resolution powerability is increased. Okay, so this is a very easy, simple question to differentiate magnification and resolution. Is this guy is equal to 57? Monotrichos, is that right? If you don't know this, you go back to look at the slides when we talk about the flagella distribution. This picture shows you very clearly the monotrichos, amphitrichos, lithotrichos, and the petrotrichos. And some of the bacteria, Pseudomonas, we will do in the lab real quick. And the Proteus vulgaris, we will always see its positive results during the biochemistry reaction. It's a very good culture to use in the